Welcome to STAT's virtual event, AI Cracking COVID-19's Code, sponsored by Adaptive Biotechnologies. Thanks for joining us. I'm Casey Ross, National Technology Correspondent here at STAT. And in just a few moments, I'll be joined by several experts to discuss how artificial intelligence is being used to better understand and combat the COVID-19 virus. We've got a lot to cover. But first, a few quick housekeeping items. At the bottom of your screen, you should see a few icons. There's a chat icon and a Q&A icon. Use the chat feature to chat amongst yourselves or to send a message directly to the STAT team. Do not put your questions for us in the chat. If you do have a question that you'd like to ask one of our featured speakers, please use the Q&A feature to type your question over to us, and we'll try to answer it live. In the Q&A window, you should also be able to see the questions that other attendees have asked and upvote the questions you would most like to see answered. We'll be taking questions throughout the chat, so please don't wait until the end to submit your questions. Thanks again to all of you for being here. And before we get started, I'd like to first welcome Julie Rubinstein, President at Adaptive Biotechnologies, for a brief introduction of today's event.
Thank you, Casey. Hi, everyone. Thanks to everybody for joining today. I hope you and your families are all safe and healthy as we continue to navigate through these uncertain times. My name is Julie Rubinstein, and I'm the president of Adaptive Biotechnologies, headquartered in Seattle. We got started about 10 years ago, and we've always operated at the intersection of genomics, big data, and healthcare. So today's topic is very near and dear to me. In fact, earlier in my career, way back in the late 90s and early 2000s, I had the opportunity to work in what was called the global e-business groups at both Johnson & Johnson and Pfizer. E-business, it was a new noun at the time. It was a word widely used across industries, but with so many different and emerging meanings. In healthcare, e-business was the start of a new era of data collection and information delivery across the industry. Fast forward 20 years ago to a talk I gave just last fall about the digital health revolution. We reflected on the incredible progress we've all made together in 20 years. We talked about the convergence of biotech and tech, the genomic revolution, the way in which data is helping segment patient populations for more targeted therapies, and the emergence of machine learning to advance research and discovery in healthcare. But we also reflected on the fun fact that e-business was no longer a noun used anywhere. The E became part of everything we do in business and in healthcare, and it basically disappeared from our lexicon. Everyone in the audience agreed that we had made so much progress and marveled at how valuable big data had become to every aspect of the healthcare industry. Today, just 9 months after that talk, it's completely revolutionized again. In fact, COVID-19 has fueled another transformation of the use of big data in healthcare. Collaborative efforts throughout the entire industry, often propelled by technology like AI, have accelerated our ability to obtain information about the biology of the SARS-CoV-2 virus, and also how people across the globe are responding to the virus. This is fundamentally altering the way therapeutics and vaccines are being researched and developed in record time so that patients can get the care they need. At Adaptive, we are passionately contributing to the generation and dissemination of immune response data to help drive solutions to the pandemic. Specifically, we are focused on how people across the globe are responding to this virus. By decoding the adaptive immune response at both the individual and the population level, we aim to make sense of the interaction between the virus and the human body, from exposure to infection to recovery. This is really hard to do, but it's a solvable big data problem if you have the right technology and the right minds behind it. In 2018, Microsoft and Adaptive forged a partnership to bring together Microsoft's machine learning, AI, and cloud computing to our immune medicine platform, accelerating our ability to map trillions of T-cell receptors to millions of clinically relevant disease antigens to which they bind. As my colleague, partner, and fellow speaker, Peter Lee at Microsoft once said, the adaptive immune system presents an extremely large but beautiful machine learning problem. Why? Because our immune cells have evolved to be massively diverse and dynamic to protect us from millions of different signals of disease that our bodies encounter every day. And each person's immune system is different, which is why people are reacting so differently to the novel coronavirus. Every single person presents the virus a little bit differently to their own immune system. And we have evolved this way as humans to ensure that no single virus or germ can completely eradicate the human race. Since the immune system sees SARS-CoV-2 just like it would any other virus, together with Microsoft, we were able to quickly mobilize our platform to map the T-cell response across thousands of people from around the world. All of the T-cell response data that we've collected so far is being made freely available to the scientific and public health communities to help accelerate solutions. At the same time that we have been generating and releasing this T-cell mapping data, researchers around the world have been publishing studies showing that antibodies don't tell the whole story about the immune response to the virus. We now have the ability to broaden the definition of immune response to include the T-cell response at scale to inform more accurate and effective diagnostics, therapeutics, and vaccines. As we'll hear today, the pandemic has spurred many novel partnerships and innovations to expedite and make drug discovery more efficient as well. COVID-19 has broken down many barriers as we band together as an industry to advance solutions as fast as technology can glean new data. Take, for instance, the global COVID R&D Alliance that is bringing together 20 of the most experienced life science and drug development companies like Schrödinger, Amgen, Pfizer, and others to identify, study, and accelerate promising treatments for COVID-19. Recently, Google Cloud also joined this alliance, donating over 16 million hours of high computing technology to advance solutions to COVID-19. Even in these uncertain times, what keeps us motivated to push forward is knowing we are all bringing our best tools and knowledge to bear and sharing what we can learn along the way. Thanks to AI and other novel technologies, today we can harness data to crack the code of disease and the immune response to disease in ways unimaginable until now. No one put it more perfectly than today's speaker, Dr. Matthew Might from the University of Alabama at Birmingham, who said, "Data is the greatest drug of the 21st century." As data continues to emerge at an unprecedented rate about the SARS-CoV-2 virus and the immune response, it's clear that there is no one-size-fits-all approach. Only by blending the best minds with the best of health and tech can we continue to make a true difference in how this and future pandemics are defeated. With that, I will turn it back to Casey and hope you all enjoyed today's discussion. Thank you.
Thank you, Julie. Now I'd like to introduce our featured speakers for today's conversation. Dr. Karen Atkinson. Karen is an Executive Vice President and Chief Biomedical Scientist and Head of R&D Discovery at Schrödinger. Peter Lee is Corporate Vice President at Microsoft Research and Incubations. And Dr. Matthew Might. Matthew is the Director of the Hugh Call Precision Medicine Institute at the University of Alabama at Birmingham. Thanks to all of you for joining me today. Also, a quick reminder to all of you out there to start submitting your questions now using the Q&A feature at the bottom of your screen. So we've got a fun conversation set up today. We're going to talk about the use of AI across multiple different research projects and sort of domains within COVID-19 research and discovery. And I wanted to start with Matt, if you could give us sort of an overview of the clinical trial, how you sort of got there with the Daguerreotypes, and how that work is proceeding.
Sure, yeah. So just to proceed immediately to the punchline and then rewind from there, we in conjunction with the National VA, launched a clinical trial of Daguerreotypes, which is used in prostate cancer treatment for the treatment of severe COVID-19 cases. The way we got there, of course, is actually a pioneering use of artificial intelligence, and I have to give a big shout out to the NCATS and the whole translator consortium upon which our funding for this AI tool is actually built. So we've created a tool called Canren, and through this consortium and through its own technology, what it can do is it can draw on many different biomedical data sets, including a condensation of the entire medical literature, transcriptomic data sets, chemical assay data sets, basically all sorts of data sets that have been transformed into knowledge, and then use kind of old-fashioned AI. And by that, I mean the more logic-driven AI than the more machine learning AI that is very popular these days, to make inferences about what might actually modulate the relevant targets for COVID-19. So you back in February when all the initial data was being released on like the genome and the targets that were coming out, we started immediately pushing them through this pipeline that's been developed and finding drugs that could modulate some of these targets. The target in this case that popped up was TMPRSS2, which does appear in the prostate cancer literature. The tool then started finding routes to modulate this using estrogens or anti-androgens. And so when we took this data to the VA, they put us in touch with the prostate cancer researchers there, and we decided on daguerreolex as one of the best routes for a randomized controlled trial for severe COVID-19 patients.
And Karen, I'd like to kick it over next to you to give us an overview of the work that you've done as part of a very large consortium of pharmaceutical companies that are applying AI to develop an antiviral.
Yes, I think as this outbreak became apparent in the US in particular, there were a group of companies who realized that we were actually all thinking about the same thing. In fact, we had been approached by a number of academics and other companies who were interested in using the technology to really speed the discovery of antivirals. And rather than compete, we decided to collaborate. And so this alliance that Schrödinger is involved in includes Takeda, Gilead, Novartis, and as was mentioned in the introductory remarks, Google Cloud. These companies came together with the idea of identifying a small molecule that could serve as an antiviral, and part of that process included evaluating the viral proteins that are used to replicate, for the virus to replicate. And the idea was really to apply our collective capabilities, which includes ultra-large screens of chemical space to identify potent selective molecules that could serve as antivirals. And so these billion compound screens, virtual screens, really powered by massively parallel compute resource, has allowed us to work on a number of these viral proteins with the goal of really rapidly, within 18 months, the idea is to come up with antivirals that we think will be the next wave of important treatments after, of course, the vaccines, which will be near term, used in the near term.
Great. And Peter, take us a little bit deeper into the work with Microsoft and Adaptive, and in particular, the role of AI in evaluating the cell receptor information.
Yeah, happy to do that. And, I think overall the response and now recovery efforts from COVID-19 have been pretty all-consuming for a large part of Microsoft. We have about 169,000 healthcare organizations around the world that are dependent in one way or another on Microsoft technologies. And so there was kind of a massive mobilization just to help that. Some of that just in very nuts and bolts healthcare response. But on the kind of side of diagnostics, therapeutics, and vaccines, besides having a donation of huge amounts of computing power for various modeling efforts, we engage deeply in a small number of collaborations. And the most important of those has been with adaptive biotechnologies. What we really focused on there is adaptive biotechnology's unique ability to analyze the genetics of T cells. And T cells really provide a well-scoped machine learning problem. While T cells themselves are incredibly complicated, if we focus just on T cell specificity, there are some important and simple principles. The most basic one is that the specificity of each T cell is completely determined by the T cell receptor. Since sequencing the T cell receptor is Adaptive's core business value, this really gave us a way to take huge amounts of data from blood samples and start to see if we could jointly use machine learning and adaptive sequencing capabilities to map T-cell receptor sequences into sets of antigens, and then analyze those sets of antigens to get to disease diagnosis. So starting about two and a half years ago with Adaptive, we started down that path. We started by redoing in a more refined and production quality machine learning and cloud-based pipeline, redoing some of the academic literature that was published in this space. Those produced very solid and promising results. And then we started to focus in on specific disease targets. We chose some infectious diseases like Lyme disease, some cancers like ovarian cancer and pancreatic cancer, and some autoimmune disorders like celiac. And so we have been making tremendous progress on this. And then when the COVID-19 pandemic hit, it was very natural for us to take the same infrastructure, the same sequencing and machine learning infrastructure, and look at COVID-19. What we've been finding is that the T-cell response sends very strong signals, and earlier signals than analyzing antibodies, and in some ways, more texture and more information in the T-cell response than you would get from just analyzing through PCR fragments of the virus. And so the data sets that we've been publishing openly for researchers and for people developing new diagnostics and new drugs, new vaccines, that's a data set called Immune Code. We're hoping will really accelerate progress to new ways to fight this disease.
Well, it's a good segue into a question I wanted to ask and kind of throw it out to all the panelists, but I'll direct it to Matt. You know, I think one of the things that is intriguing to a lot of people is the use of AI and the progress that's being made during this pandemic. Why is AI proving to be more effective, perhaps, in seeking answers now versus prior pandemics where we didn't see that level of investment or focus on it?
Yeah, I think part of it is that AI as a field has evolved rapidly, really over the past 10 to 20 years. Even though we rely in large part on sort of old-fashioned logic-driven AI, and maybe in part because that's sort of coming back and has been so useful. I just think we have far more tools at our disposal now, and also far greater computation. One of the lessons of machine learning is that the more compute you have and the more data you have, the better you can do.
Yeah, just to jump in there on the compute, in Microsoft Research, we have a GPU hypercluster, and we were able to donate close to 30 petaflops of compute to various vaccine and drug development efforts, and mainly for molecular and thermodynamic modeling, but there was a lot of machine learning there, including machine learning that we're doing with adaptive biotechnologies. Yes. One stat I've used in the past is, there was one of the vaccine candidates from Immunity Bio that for about 4 weeks, they were consuming 24 petaflops continuously of computing power. And that 24 petaflops is roughly 200 times bigger than the largest supercomputer on the planet back during the first SARS outbreak. And so it's just night and day, the amount of computing power that's available. And the data maybe has advanced even more.
Yeah,
I'm sorry, go ahead, Karen.
I was just going to add that in the design of these molecules, we leverage physics-based methods, which really are able to capture the complexity of molecular properties and interactions between molecules. But those are computationally very expensive simulations to run. And so having access to machine learning to really extend the reach of these physics-based approaches has been really tremendously beneficial. And to sort of, again, give some scope to what we're talking about, the 16 million hours that was donated by Google to perform these calculations, that equates to about 1,826 years worth of calculations. And that's sort of hard to comprehend how we could have done that a decade ago. So really exciting that we have now at our disposal this amount of compute power that really, I think, augments our ability of scientists to work on these difficult problems.
Yeah, I just want to add again that the filtering effect is actually really important, so that it focuses where you're searching, which can then be validated with other more in-depth methods. So we use AI as a filter and then validate with cell models or animal models.
And Karen, I had a question that was submitted for you. A listener wanted to understand a little bit better the general business agreement among the members of the alliance you're working in. Can you give a quick overview of that?
I think I can briefly say that, as mentioned, the idea here was for many companies to come together with their synergistic capabilities. The idea really behind this is to put these capabilities into this joint project and make the outcomes, the small molecules that we identify, available. So this is a philanthropic effort. I think that's important to point out. The idea is really to put this information out into the public domain ultimately for clinical development.
And another question that's come in, and I can direct this one to Matt. Is anyone working on blocking ACE2 receptors in the lung? Isn't that where COVID-19 attaches to the body? Is that, do you have insights into that, Matt?
Sure, and I think Karen probably has some thoughts on this too. So the spike protein on the virus docks to ACE2 in lungs, and so sort of the immediate rational strategy that emerges is, can you in some way inhibit that interaction between ACE2 and the receptor binding domain on the spike protein? Another strategy we initially considered was, can you just decrease the amount of ACE2 in the lung to reduce the number of binding sites? And we quickly realized that this could be problematic for people for a number of reasons, which I won't get into right now. But certainly we have looked at approaches to finding ways to block or to stick to the receptor binding domain in some way. The problem is, the binding between the spike protein and ACE2 is very, very, it's strong. And so to compete with that is actually quite difficult from a small molecule perspective. We haven't found anything sort of so far that way that will work. So we've looked at other targets in the interim. So Karen may have other comments on this.
I think your last comment, I'll just add to say that interaction is quite strong. And I think also the size of the interaction between those two proteins is significant. And so I believe there are antibodies that are being explored that could potentially disrupt this interaction. It's not something that we are working on though.
A question I wanted to direct your way, Peter. A listener is wondering if you can elucidate more on how AI is helping with selecting the viral strain for the development of the COVID vaccine.
Yeah, so one in the pipeline with adaptive, you can think of the machine learning as being in 3 stages. Once a Tcell binds to the virus, it undergoes a process of clonal expansion. And so when you take a blood sample, one of the first problems you want to do is to identify those T cells that have recently undergone that clonal expansion. And that ends up being a first machine learning problem. Then secondly, once you have that, then you want to go through a process of mapping those T cells to antigens. And that mapping process structurally has similarities to what we do in machine translation. So, for example, if we're trying to translate, say, between English and Mandarin. And in fact, interestingly, it's of a size that is on the same order of magnitude as translating natural language. So it's very challenging and large machine learning problem, but we can actually get some useful results of that today with today's capabilities. And then once you have that, then there's a kind of problem that is very similar to the machine learning problem of topic identification on the web in order to basically provide a precise diagnosis. So that pipeline basically ends up producing not only diagnosis, but along the way, you produce data that's now codified in this immune code data set that has a few things, hints in it, we think. Besides just a clear diagnosis with very high specificity and sensitivity from very early onset of infection, it also provides a different picture of the response of individuals. It may provide a basis for stratifying individuals for which vaccine candidates might be most effective. It might even provide predictive power, even pre-infection, on likely response should a person get infected. And so all of these kinds of elements in the data end up being produced along the way through this machine learning pipeline. And we've tried our best working with Harlan Robbins and his team at Adaptive Biotechnologies, and then Jonathan Carlson and his team here at Microsoft to provide in this useful form all of those data products open to all researchers. For vaccine and drug development, then, we think that there is a further use for this data in really trying to understand much more precisely how people are responding to these vaccine candidates or to drug therapies. And so it really, while the whole pipeline is really focused on giving you a very precise, highly sensitive, highly specific diagnosis, the data products along the way provide just a really detailed picture of T-cell response to COVID-19.
And I think a good general question that has come in, and I wondered, Matt, if you could take this one on, and then maybe Karen, if you could jump in here. A listener's wondering sort of what bottlenecks that you've encountered in collecting and sharing data. It seems like there's been a lot of good collaboration during this time period, but what are some of the barriers you're still sort of facing?
Oddly enough, I mean, it's sort of been the ultimate bottleneck breaker, if you will. So as part of this NCATS Translator Consortium, we put out an urgent plea to get more of the rapidly emerging COVID literature into the knowledge graphs that we all share. It happened really fast because people understood the urgency of that. And then UAB is part of this, national COVID cohort collective where we're all merging our EHR data. That would have been unthinkable. I mean, just completely unthinkable. And then we're starting to do, machine learning and data mining on top of that data to look for differences in survival and mortality. Trying to think of what bottlenecks haven't been broken by COVID, because there's ample compute now. There's ample data. The one that I guess hasn't completely fallen yet is that there's the literature is still locked away behind paywalls. So some of the interesting papers that we needed to get to actually were still behind paywalls. So for example, a lot of the prostate cancer literature that we needed to validate this TMPRSS2 idea was locked up. So that still hasn't fallen. That's still in place.
Yeah, that's actually an interesting issue. With 1 million papers being published every year, that's really imperative that scientists are able to access all of those papers. But on the question of sharing data, I think this collaboration that we're involved in is unique in a way. It's many companies who usually work separately and don't really share a lot of data, really trying to come together and share information about projects, about compounds in particular. And so that, I think, is a sort of new experience, if you like, that we're working through. What's great is, I think as part of this collaboration, one of the tools that we use at Schrödinger called Live Design, it's a sort of collaborative tool that allows scientists, no matter whether they're chemists or biologists, to really put all the data in sort of one space. That, I think, has been very useful. We've seen actually a lot of interest in that tool because everyone's working remotely. Where can we store all of this information, not just through the collaboration, but more generally, so that we can design molecules from literally our homes. That has been quite useful, I think, so far in the collaboration. There are still some things to work through in terms of sharing compounds and so on. But I think, traditionally, a lot of systems across different companies are different. And that makes sort of interoperability or interconnection of data pretty challenging, and it's not something we've really had to deal with a lot in the past. I think the opportunity here is that we figure out some of this in the context of COVID-19, and that makes collaboration easier going forward. I think one of the things that I'm hoping comes out of this is that if we can figure out how to work together on this urgent situation of COVID-19, when it comes to antibacterials, antifungals, some of the real huge unmet need there in terms of other infectious diseases, that some of what we've learned here can then be applied going forward in more of these collaborative endeavors across companies.
Just to add to what Matt and Karen said, I think early on in the pandemic response, the tech industry was actually, with all good intentions, was racing ahead and creating yet more data silos. But I think as time has gone on, we've sort of gotten a grip on that, and the pooling of data and authentic thoughts about standardization of that data is actually really interesting and exciting. And to Matt's point about EHR data getting pooled, that, at least from where we're sitting in Microsoft, that's now starting to expose the true realities of real world evidence kinds of investigations. And I have to say, I've been guilty of thinking while data was harder to get, that the concept of real world evidence, of combining genomic data and clinical records and doing machine learning, I thought was relatively straightforward, but we're now learning, actually it's very subtle and much, much more difficult than we thought. And those learnings, I think, are so important to progress in the future. So while I have to say that our team gets stumped more often now than just a year ago, I view that all as a good sign, because it really focuses our attention on probably the really important problems to work on that we just weren't working on a year ago.
And a question to you, Karen. A lot of a listener writes that there's a lot of false hits on docking studies recently. Wondering your perspective on what's causing those, what needs to happen to make those predictions better.
So I think there's a sort of spectrum in terms of what one is able to do with a crystal structure of a protein. One of those things is docking small molecules. The other, though, is working at the atomistic level on these physics-based calculations. And I think if you open many journals these days, you will see, beautiful pictures of compounds docked to proteins. I think that's a level of resolution and rigor that is open to error. And that's why we're so focused on these physics-based calculations, because frankly, if you have the wrong interaction or the wrong docking model, it shows up in the calculations. And so that's one of the reasons why we're really focused on that, and that it is really the foundation of how we think about using structure-based drug design. So I think some of what we read in the literature is really because people aren't necessarily leveraging that level of atomistic interaction and modeling.
Yeah, if I could just add to that and add my own experience with the listener's question. We started getting into docking, I guess, around March and April with this. And suddenly got very excited about all these polyphenols that kept showing up and binding to all these targets. Then we realized that, well, polyphenols just bind to everything. they're very promiscuous molecules. And we just sort of rediscovered what I guess was already widely known in the docking and physics-based simulation community. But they didn't stop these molecules from getting a lot of attention on social media, things like quercetin and stuff like that, and having people think that they could go out and buy enough dietary supplements. When you finally work out just how much you'd have to take of these things to actually reach the concentrations where you might inhibit some of these bindings, it's absurd.
Right. And I think that's really the focus of these drug discovery programs in this collaboration, is to come up with potent, selective molecules that can be safely taken by large groups of people who unfortunately may become sick with the virus.
A listener points out that AI is a very general term, as I'm sure everybody here can appreciate, and wanted to get a better sense of the AI techniques that have proven effective in each of your efforts. So maybe we can go right down the line, and Peter, you can talk a little bit more about the type of AI you're employing and how it's proving effective.
Yeah, it's, I think, well, first of all, when we're talking about AI, we're generally talking about machine learning and machine learning for various classification, ranking, or reading or, analytics problems. And then within machine learning, there is generally speaking, the thing that we've been doing with adaptive biotechnology has been focused on various deep learning techniques. There is a boosting process there. And so one of the things that we do is we start with a relatively small cohort of affected individuals and the blood samples from those, but then boost the models with a very large number of naive samples. And the reason that that works is that all human beings have a large number of T-cells that are specific to all sorts of diseases just through the randomization of the receptor as it gets produced in the thymus. And so, so Casey, you and I both likely have T-cells that are specific to ovarian cancer, for example, even though you and I aren't susceptible to that disease. And so with a little bit of biological information and a large number of samples, even if they're naive, we're able to use fairly standard machine learning techniques like boosting. And ultimately, the only thing that's truly unusual about the machine learning is just the scale of the problems. They end up being, roughly speaking, web scale. One last thing is, and this is something Karen and Matt referred to earlier with publications. Dealing with over 1 million publications a year is difficult. In fact, just in oncology alone, there are about 2,000 publications per day. And so for cancer treatment, for example, for a tumor board to make decisions, there's just no way for a human group, in a tumor board to be up on all of that literature. And so whole document machine reading and problems like this that are just on the hairy edge of what's practical today are really starting to emerge very rapidly right now. And so we start to see moving away from the classical kind of deep learning to more reinforcement learning, more auto ML techniques. Those things are just starting to get deployed in more and more situations, and it's actually remarkably comprehensive what's being applied now. I'll bet Matt and Karen have a lot more to say on this as well.
Yeah, Matt, why don't you get on the machine learning train, my friend?
That's right. So yeah, in terms of what we're using, I mean, we use a full spectrum of AI technologies. We're probably a little bit different in that a big portion of our portfolio really is more old-fashioned logic-driven AI. This is the stuff that was popular from the 60s up through the 80s, where you really are combining sentences using rules of inference, trying to deduce new knowledge. And there was a time when people thought that that could ultimately, in the limit, approximate human intelligence. You might approximate Vulcan intelligence that way, but it doesn't give you a human being. Still, it's very useful for deducing non-obvious facts from the literature by combining disparate pieces of knowledge together. So on the newer AI side, we do rely a lot on natural language processing to soak up information from the literature. We're in many cases restricted to just the abstract that's available on PubMed, but we do what we can with it. The translator consortium that we're a part of, which provides a lot of the data sets that we use, uses probably every AI technique in the book to produce the various data sets and data minings that they do.
Maybe just to add to what Peter and Matt said, there is a difference, I think, that needs to be established in terms of what one does with machine learning when you're looking through existing data sets, in particular compounds, but I think it's true of all data sets in some way, that looking through existing data sets can be really powerfully impacted by machine learning to extract key pieces of information. Again, when we think about the use of deep learning, because we're really looking for molecules in chemical space that may not have found their way into existing libraries, we rely first on creating an understanding of these interactions, these molecular interactions, and using machine learning, in this case deep learning, to work our way through larger amounts of chemical space. So we're not really sort of using existing chemical libraries to look for those molecules. We're looking in chemical space, literally using deep learning and these physics-based methods. I should say I'm not a computational scientist or a computer scientist. I'm a translational scientist, and I think if Robert Abel, our head of science, were here, he'd tell you a lot more about the specific types of AI and deep learning, machine learning that we use. But I think really looking in chemical space does require the combining of the accuracy of physics with the speed of machine learning.
And staying with you, Karen, for a moment, a question that came in from a listener that I wanted to add to a little bit. The question was, is there a meaningful challenge? Are there meaningful challenges that arise from mutations in this virus? And what I'd add to that is, if so, how is the AI able to sort of help you tackle those?
So I think that understanding is ahead of us. I think there's a lot more to learn about mutations. I would say just in general, the use of these approaches allows us, in some cases, to simulate what a protein structure looks like in the setting of a mutation. So let's imagine we have the wild type protein. Typically and historically, we would express that protein and we would look for differences on the biochemical or cellular level in terms of the function of that protein. I think what these methods allow us to do is really to simulate that on the computer. If you change a single amino acid in that sequence, that's going to have an impact on the three-dimensional structure of the protein. And so what one can imagine is the ability to mutate residues in silico to understand what that does in terms of binding, for example, of a molecule, or in other cases, it would be binding of an antibody. So this sort of in silico mutagenesis of a protein is something that I think is a potential application in COVID-19. It's clearly being used in other applications like oncology, and that's something our teams leverage. So I would say that yes, there is potential here. I don't think we know enough yet about the mutations in COVID-19 to be able to apply them directly to the projects we're working on right now. But as more data becomes available, I think there's definitely that possibility.
I'll just add that we are looking at mutations that could occur to evade some of the potential therapeutics we're thinking about. So, for example, if we successfully inhibit TMPRSS2 to the point where it no longer cleaves effectively, what mutations might enable other proteases to cleave effectively? So we're looking at stuff like that too.
And a question for you, Peter. A listener wonders if, from the Tcell response changes that you've studied, come to light as part of your research, does any of that shed light on why older adults are just more likely to get this virus and suffer from it?
Yeah, so there are differences. One thing, at least in the current ImmuneCode data set, one of the things that has been a curiosity for us is to look at differences according to age and with a particular focus on small children versus adults. The current ImmuneCode data that we're collecting does not have that today, but we are starting to see some stratification according to age, people over 60 and versus under 60. And those are actually called out in the immune code data set. I think, as I'll use a phrase Karen just said, I think our understanding of what this means is still ahead of us. But one way to think about it is that you can actually look at some of the T-cell response and make a prediction of the person's age. And so there is some, there is something going on there that we don't fully understand. And this is another hint that maybe there is some predictive power, even in kind of pre-infected people, that might give some clues as to who's likely to get seriously ill and not. And so again, these are just all questions of intense interest for us, and we think they may be lurking in the data, the answers.
And a couple of people have raised the question about whether machine learning algorithms will be made sort of publicly available, in an open source sort of way. And I wondered if you can comment on that. Karen, maybe just talk a little bit about your project in particular, whether those, codes will be shared.
So some of the underlying software that's used in the design of molecules across the Schrödinger suite, those are actually available, I believe, for download for academic use, and schools use some of the software as well as industry. So that some of the data and some of the methods we use are actually broadly available. The use of them in specifically these projects, I don't think there's any change in the software and the way we use these algorithms. We do intend to broadly make available, though, the outcomes of this research, and I'm imagining some of the ways in which we've applied the technology, and not specifically machine learning, but the ways in which we've applied the technology to come up with these molecules. Those will be put out into the public domain, I'm sure, as publications and scientific presentations that will allow people to get insight into, just how this works.
And a question for you, Matt. What are the sort of most exciting developments that you've seen that, sort of were accelerated by COVID-19, from a computer science perspective?
So from a computer science perspective, I would say it was a chance to finally validate a lot of what's been brewing for some time. And also the way it's brought computer science together with biologists in a very serious way. I had a lot of people reach out to me from my old computer science life and say, hey, how can I help? And I thought that was interesting to see. So not at a technical level, but sort of at a human level, to see so much interest coming from the field. And actually, I believe just in general that what's been missing in medicine and biology for a long time isn't more computer science; it's more computer scientists. And if this is the event that brings them over, that's really exciting. For me personally, it's also been exciting to see that just because logical AI is old doesn't mean that it's not useful. It's actually been tremendously useful to wake up this old technology, spin it up to a scale it's never been used on before, and see what it can do.
Peter.
I totally agree with Matt, and I would add that we also get to face reality a little bit. There's always been this dangerous, irrational exuberance. People in healthcare look at the computer scientists with awe and all this magical machine learning power, and then people in computer science, particularly machine learning, look at a lot of problems in healthcare and biology and think, we should be able to tackle those pretty directly. But it's actually a little bit kind of over-optimistic in both directions, and that actually creates some dangers because both sides then come together with a little bit of irrational exuberance, and mistakes get made. And so I think I just see a lot more grounded realism in reality. In the kinds of collaborations taking place right now. And, just from a learning perspective, people are learning really fast right now. Just the amount that's been learned in the last 12 weeks is at least 12 months or maybe even 12 years worth of sort of cultural lessons.
Yeah, I think if this colonization of medicine and biology by computer scientists lasts, it'll have a long impact on the field.
Yeah, and I'll just add to that in saying that coming from a traditional 25-year career in pharma and joining a software company, I've been able to see firsthand how those two worlds interact, and I, for one, am very excited about the potential there. I do think, as Peter just said, we have to be realistic. This isn't magic. We're not going to be able to cure diseases with AI per se, but what we can do, and the other sort of term for AI I use is augmented intelligence, right? It's going to augment the ability of chemists, biologists, pharmacologists, who essentially have to do a lot of the work after we identify molecules using these acceleration methods. The pharmacology still needs to be done. We still need to test these in people, and certainly machine learning, I think, can help there. But it's really the coming together of these interdisciplinary teams that I think is very, very exciting to accelerate our work and also to make more headway collectively across a number of projects.
Actually, one thing to add there, because Karen has brought up several times, the importance of bringing in knowledge from physics and chemistry and biology into this. And that's really important because just with today's machine learning capabilities, which are very data dependent, there's always going to be this lack of data for some of the problems that we want to solve. One way I've tried to explain this to my fellow computer scientists is that since the beginning of Homo sapiens, there have only been roughly 106 billion people who have ever existed. And so even if we had genetic samples from every single human being ever, in a computer science perspective, it's a relatively small amount of data. And so just 106 billion genomes. And so for some of the problems that confront the worlds of biology and medical research, we're always going to be data constrained. And so we need a deeper kind of physics and biology understanding in order to make real headway.
How much priority is placed on finding molecules to antagonize the virus or inhibit its binding versus simply controlling the individual's immune response and modulating that to prevent the hyperactivity, which is a significant contributor to mortality? Matt, can you take that one?
Sure. So there's a lot of energy going into both, I would say. For us and for a lot of folks, it was easier to start on the antiviral side, just because we had the sequence, we had some of the structures, and we could apply a lot of the classical tricks. The immune system is really complex, and modulating its behavior is not easy to do. Our work had to really await for the arrival of the higher organisms like the ferret model to start validating some of the predictions there. So it's just taken longer to get to the point where we're looking at predicted therapies to modulate the immune system in a favorable way. I mean, we made predictions at about the same rate for both camps, but the validation occurred, in the biology much faster for the antiviral category than the immune category. What's actually really helped on the immune side is getting access to large amounts of EHR data. So that's what's really giving us a sense of what might truly work in humans when it comes to immune modulation. But only now is that data arriving at the scale where we can start to make statistically confident inferences about what we're seeing and propose, based on that, randomized controlled trials that would validate these ideas.
And we're about out of time, but I sort of wanted to ask one last wrap-up question and kind of go right down the line if we can, and have each of you answer it. And this has to do with sort of the ability to translate the research that you've all been doing, get it into human beings and make a difference. It's proceeding at a pretty rapid pace, I would say, but, maybe starting with you, Peter, the ability to develop the kind of tool that you're trying to develop and to get it validated to the point where, you're confident deploying it.
I think that we are in a new world, and so there are early access programs and emergency use authorizations that are being pursued for several things. I think a lot of the issues are actually more operational and logistical. And so really focusing on how do we get, once we do get vaccines that are safe, how do we deploy them? And if they require multiple shots, how do we track that people are getting those safely? And so that's where it's just a lot of nuts and bolts IT work, coordinating with companies like Walgreens and Walmart, CVS, Kroger Health, and so on. And so we're putting a huge amount of effort into those things, but it's going to be, once we get these things deployable, it's going to be a massive global effort to get things deployed as quickly as possible to people who need it.
And Matt, in your realm, the work you're doing, the clinical trial, tell us how that's proceeding, and sort of what you hope to see in terms of timing and being able to apply some of the things that you've discovered and help people.
Yeah, so we were able to put the protocol together in April and get it funded and running by early May. So we should start to see the data come out of that sometime this fall. What we're certainly hoping to see is that in severe patients with COVID, that daguerreolex was effective at improving survival. That's what we want to see. But we're also going to look at things like the length of hospitalization and some of the things that might make a difference in the overall scale of the pandemic.
And Karen, realizing that it's human beings, not necessarily AI that makes these discoveries. AI is certainly accelerating the work, but sort of give us a sense of timeline on your end about when some of the discoveries that you're making sort of might begin to translate and impact care.
So the thing I think about this multi-company effort is that you have a number of capabilities at the disposal of the collaboration. As we've said, this work that we're doing now is about accelerating the pace with which we can discover these molecules that we hope will be safe and effective in as many people as possible. Following that period of discovery comes the clinical development. And I think that the goal of this collaboration is that within 18 months, we will have identified antiviral therapies that can go into clinical development. So this is, again, not about vaccines, which are the first wave of treatments, and not treatments, but first wave that we hope will have huge impact. But following that and on an ongoing basis, we think antivirals are going to be very important in managing this over the many years. And so we hope that the acceleration in discovering these molecules, that we'll be able to benefit collectively around the acceleration that other groups have been able to pull off in terms of clinical trials and working with regulators. I think that's all, again, ahead of us, but our aim is to shorten that time that it takes to get something ready for clinical testing.
Well, with that, sadly, I think we have to wrap up. And I hate to say that because there are so many questions that came in that we just simply weren't able to get to. But, I want to say thanks again to our event sponsor, Adaptive Biotechnologies, and thanks to our featured speakers for joining us and for all of your efforts in COVID-19. And thanks to all of you out there for attending. Please keep an eye out for some more STAT virtual events happening soon. And until then, stay safe and be well.